Children, you understood about the dicot root. This is just a representation of this dicot root. Please don't learn this diagram. It is only for the explanation part. So it will be easier for you to understand if I do this in circles. Okay, that is why I am showing this. Don't read this diagram or don't draw this diagram in your examination. Only the diagram given in your textbook should be done. I am just saying about this different parts of the dicot root using this diagram. You please don't use this diagram to do in your examinations and all. Okay, so let us see about the different parts of the dicot root. Okay, so first let us see the outermost part. So what is the outermost part? It is the epidermis. Okay, the outermost layer is the epidermis, and it protrudes and this epidermis will protrude out in the form of the unicellular root hair. We know that we are talking about root, right? So epidermis gives rise to the root hair. So that is called as epidermis. Next to epidermis is the next layer, which is called as cortex. So this part completely, it is the cortex, right? So here this line, starting from this line, this circle till the epidermis, it is called as cortex, right? And what does this cortex include? It includes the parenchyma cells and also the endodermis, right? So cortex includes parenchyma cells and endodermis, right? So we will see how the parenchyma cells are arranged. So parenchyma cells, there are several layers of thin-walled parenchyma cells with intercellular spaces. You can see this layer, it is completely surrounded by the parenchyma cell. I am not drawing it. I'm just saying that it has many layers of this parenchymatous cell. Completely the cortex is surrounded by the parenchyma cell, and these are thin-walled. See, I am not drawing it completely. Just understand that space will be fully covered by thin-walled parenchyma cells with intercellular space. With intercellular space means there will be space between the cells. Okay, and let us see the next part of the cortex, which is the endodermis, right? I told you parenchyma cells and the endodermis together forms the cortex, right? So what is this endodermis? So this circle which you see is the endodermis, and this endodermis is a single layer of barrel shaped cells. I hope you remember what we saw in the PowerPoint presentation. So these are the barrel shaped endodermis, and they are single layered cells. You can see it, I have drawn only single layer, right? So these are single layered barrel shaped cells without intercellular space. You can see there won't be any space between the each cell of the endodermis, right? And here in endodermis we are able to find a special type of stripes which is called as the Casparian stripes, right? So what is it? Casparian stripes. And where are they found? They are found in the endodermis. Now what is this Casparian stripes? Stripes എന്ന് പറയുന്നത് normally വര എന്നുള്ള ഒരു അർത്ഥമല്ലേ. So here in the endodermis, in their tangential and also in the radial wall. Tangential എന്ന് പറഞ്ഞാൽ അതിൻ്റെ മേൽഭാഗത്തുള്ള ആ ഒരു സർഫേസിലും അതിൻ്റെ radial wallsലും, this part, right? So radially their walls have a deposition of water impermeable. Notice water impermeable. Water cannot enter through this layer, right? So it has a water impermeable waxy material which is called as suberin, right? So this tangential and radial walls, they have water impermeable waxy material which is called as the suberin, right? So upon the endodermisന്റെ മുകളിലാണ് നമ്മളിത് കാണുക, and അവരെ radial wallsലും ഓരോരു സെല്ലിൻ്റെ റേഡിയസിലും we will be able to find this Casparian stripes which is made up of waxy material which is called as the suberin, right? So this is about the endodermis. Now coming to the next part, which is the pericycle. You can see the next layer nearer to the endodermis which is called as the pericycle. Now what is this pericycle made up of? It is a few layers of, I have not shown all the layers, it is a few layers of thick-walled parenchyma cells. So cortex will have thin-walled parenchyma cells and pericycle will have few layers of thick-walled parenchymatous cell. And it is the point of initiation of the lateral roots and vascular cambium during the secondary growth which takes place in these cells. അപ്പോൾ ഈ ഒരു പെരിസൈക്കിൾ എന്ന് പറയുന്ന thick parenchymatous cells-ലാണ് when there is a lateral root formation. Okay, that is lateral roots എന്ന് പറയുന്ന roots which arise later, right? So these lateral roots and vascular cambium are formed from the pericycle during the secondary growth when it takes place in the cell. Right? Clear with that? Okay, now let us move on to the next part, which is the pith. Okay, so what is this pith? Pith is the small portion in the center portion of the root system or stem system. We are able to find a small part which is called as the pith. Okay, so pith, it is small and it is inconspicuous. It means that either it will be very small or else we won't be able to see this pith at all in the case of the dicot root. Okay, and then let us learn about a special type of tissue which is called as conjunctive tissue. What is it? Conjunctive tissue. Okay, so this conjunctive tissue is present between the xylem and phloem. So in this diagram, the red indicates the xylem. I will just represent it. It is xylem, and the blue color, it represents the phloem. Okay, and what are these conjunctive tissues? These are nothing but they are parenchymatous cell. You can see yellow colored cells which I have drawn, right? So in between the xylem and phloem, there are many parenchymatous cell which we call it as conjunctive tissue, right? So this is about the conjunctive tissue. So it lies between the xylem and phloem. Okay, and now let us learn about xylem and phloem itself. So there will be 2 or 4 xylem and phloem. Okay, you can see it here. It is 1, 2, 3, 4. So phloem is 4 and same way xylem is also 4. 1, 2, 3, 4. Okay, so either it will be 2 or 4 and they are found as patches. You can see it is found as patches around the endodermis. Okay, later what happens? A cambium ring will develop between xylem and phloem. Okay, so later a cambium ring will develop between the xylem and phloem. Understood it? So all the tissues on the inner side of the endodermis such as pericycle, vascular bundle and pith constitutes the stem. Okay, clear with the children? Clear with dicot root? Okay, now let us learn about the monocot root. It is very easy. If you are clear with the dicot root, you can easily study this monocot root. Okay, so it includes epidermis, cortex, endodermis, pericycle, vascular bundle and the pith. So all the things which we saw in the dicot root is seen in the monocot root also. Okay, it is seen in monocot root too. Clear, children? So it includes the epidermis, cortex, endodermis, pericycle, vascular bundle and pith. Epidermis, it is outermost layer and you can see the cortex layer till this. So these are thin wall parenchymatous cell which have intracellular space, and then we see the endodermis, and then inner to us is the pericycle, and then comes the vascular bundle and the pith. Okay, now there are usually more than 6 polyarch xylem bundles. So how many xylem bundles will be there? There will be 6 xylem bundles in them, right? So in the dicot root we saw that the xylem were found and xylem and phloem were found in different radius, but here it is seen in the same radius. Okay, xylem and phloem will be seen in the same radius. You can see it here. This is the xylem, which includes the protoxylem and metaxylem, and then you can see nearer to it is the phloem cells, right? So how are the xylem? They are usually polyarch, and that is they will be either 6 or more than 6. Here they have shown 8, right? So they are polyarch, that is more than 6, and the difference which we find here, other things, everything is same, that is epidermis, cortex, endodermis, then the pericycle, everything is same, but only difference is they will have more than 6 xylem bundles, and the pith will be large. In monocot root, we are able to find that the pith central portion will be large and it will be well developed. But in the case of dicot, we saw that the pith were small, or it was inconspicuous. Okay, and they don't have this conjunctive tissue in between the xylem and phloem, and along with that they do not show any secondary growth, right? So secondary growth is not seen in here. That means that the lateral roots and the secondary growth will not take place in the monocot root. So other than that, everything is same. Only the difference which we have to study is they are usually more than 6. That is, xylem bundle will be seen more than 6, and pith is large and well developed and no secondary growth. Okay, so this is the difference between monocot and the dicot root, children. Now let us move on to the next part, which is the monocot and dicot stem. Okay, I hope you are clear with that. Read it properly. If you have any doubts, you should ask to me. Okay.